everybody. So now let us continue the discussion further. Today we are going to talk about the relevance of intermolecular interactions, non-covalent interactions in supramolecular processes related to biological systems. And in this regard, we are going to look at a very important classification of compounds which are called enzymes. And we know that enzymes are very important biological molecules that function as a very active catalyst that bring about suitable reactions under the right conditions at extremely fast and accelerated rates. So enzymes are essentially the workhorses of molecular systems, where they catalyze different chemical processes with unprecedented precision at very fast rates, and the ability to carry out this process is millions of times without any loss in activity, and also these enzymes they function under physiological pH and room temperature conditions. So this makes naturally occurring enzymes extremely efficient. And so what the chemists were thinking is that can we actually make some systems, some specific systems where the chemical reactions which are performed in such systems, they model the enzymatic processes, and whether we are able to replicate the same level of efficiency that is happening in a naturally occurring enzyme. So with this background in mind, chemists got into the process of making specific molecules. For example, we have got a biological receptor, and then we have got a substrate molecule that is going to, or a set of substrate molecules that is going to bind to the biological receptor, and then there is going to be a transition state like a situation created where the reacting molecules come close to each other, they recognize each other, the chemical reaction takes place, the products are formed, and then the products are again released back into the solution. And this process where the product formation takes place at unprecedented rates is what makes these biological workhorses extremely efficient. So chemists came up with the idea of trying to design enzyme mimics, where we have got enzyme mimics, where we have got, say, a enzyme which has got a particular shape, and then we have got, say, we have got two reactant molecules. Okay, for example, we have got two reactant molecules A and B, and the idea is that they interact. These are called the substrates, and this is the enzyme which is called the receptor. And the enzyme is going to interact with the substrate via non-covalent interactions, and the shape, there is a slight conformational change so as to accommodate A and accommodate B. Then there is a chemical reaction between A and B to form D and C, and finally the decomplexation takes place. This is the complexation process. This is the reaction site, and at the reaction site we can see that the A and the B molecules come very close to each other, and they create a transition state like situation. So essentially they mimic the transition state such that the reacting partners are extremely close to each other, the chemical reaction takes place, and then the products C and D are released into the bulk. And this is what is going to be the topic of the current discussion, that is, we are going to look at enzyme mimics. And before we go on to that, let us look at the fact that we are trying to develop a structural model that mimics enzymatic activity. And the reason for the enhanced enzymatic activity is the lowering of the activation barrier, that is the free energy associated with the process, to a pretty reduced value, such that the reaction rates can now happen at very fast, and the reaction can take place in the shortest possible time. So we need to mimic the enzyme activity, and we need to develop structural models for that. However, we need to keep in mind that naturally occurring enzymes which function very efficiently have got very precise structural, binding and catalytic feature. These have got very precise structural, binding and catalytic features, and it is not easy, or it is in other words very difficult to incorporate all the structural, binding and catalytic features into a given structural model, and that is where chemists have been trying to design suitable structural models that can mimic the enzyme activity. And some of the most important features of enzymes, as I told you, is fast reaction rates under mild conditions. That is, unlike traditional organic reactions where you can employ very harsh conditions, like the use of high temperature and use of stoichiometric amount of catalyst, and you use reagents to bring about transformations, and yet the rates need not be the fast rates. The reaction can still be sluggish or proceed at moderate rates. Enzymes, they have fast reaction rates under mild conditions. There is a very high degree of structural recognition of their substrates. So you can see here that although this particular region was not really having the right match for this particular substrate, there is now a change in the conformation of the enzyme to accommodate this reacting molecule, so that the structural recognition which happens between the enzyme and the BA reactant happens with the highest degree of molecular recognition. The third factor, as I told you, there is a very high turnover number. So these enzymes, they catalyze the reaction of a large number of substrate molecules with high precision without being destroyed itself. So very high turnover number, and large number of substrate molecules are catalyzed at very high levels of precision without being destroyed itself. And there is also another important process which operates. There is competitive inhibition by compounds that bind to the enzyme but do not react themselves. So this is also a parallel process which operates, that is, you can sometimes have a substrate which is able to bind to the active site of the enzyme, but actually does not have the necessary predisposition of the functional groups to react with the other reacting partner. So this particular competition, or which can actually inhibit the enzymatic activity, also operates in parallel in the case of enzymatic processes. So this factor also has to be kept in mind. Now, so when we need to have the design the structural model, what are the prerequisites in the structural model? prerequisites for a structural model. And so to start with the structural model, it must be analogous to the transition state of a biological reaction when the substrate binding happens with the system of interest. So, it must resemble the transition state for a particular biological reaction when the substrate binds with that system of interest. And an intermolecular system, that carries out the reaction concerned without the binding characteristics of the enzyme, and this is called the functional model. So, there is a functional aspect and there is a structural aspect. Both these aspects are very important when it comes to the design of the requisite structural model. And also we need to keep in mind that unlike biologically occurring natural enzymes, where the rate of the reactions are extremely fast and they occur in a very highly efficient manner, the biological mimics or the structural mimics that we are going to make to model these biological processes will definitely happen much more slowly in comparison to that of a natural enzyme with a substrate. So, furthermore, what we should have is the hydrophobic binding site for the substrate, hydrogen bonding or electrostatic interactions, that is binding sites that are complementary to the substrate, catalytic groups must be attached to this model. The structure must be well defined and rigid, and finally the model must be water soluble. This is extremely important, and catalytically active under physiological conditions. So, these are the most important properties that must be present in the structural model, which are mentioned here, and the last 5 are extremely important. We must have suitable hydrophobic sites, we must have electrostatic binding sites, catalytic groups must be a pretty rigid and predefined structure, and water soluble, and be catalytically active under physiological conditions. So, with this background in mind, let us try to look at some of the relevant examples that are present, and in general we would like to emphasize the fact that now when you have got the enzyme which is not now recognized the starting materials, the reactants, so the entire reaction process has to happen in a essentially within this unimolecular species, which now contains the reactant molecules as well as the enzyme itself. So overall there is one large large molecule, and which positions the reacting species in close proximity, so that the chemical reaction takes place, products form, and then the decomplexation happens such that the final products are now released into the bulk. So, in this regard, it is the intramolecular process which takes place with the enzyme that is known to proceed with greater selectivity compared to the intermolecular processes. So, let us now emphasize this fact that intramolecular reactions proceed faster than the biomolecular equivalents. So, unimolecular reactions, they proceed faster than the biomolecular equivalents. For example, let us consider this reaction. So, here we are going to have a nucleophilic attack at the carbonyl center, and then this is takes place with the rate constant K1, and this is going to form this product plus, so we have a X here, and then the hydrolysis of this is going to give me the dicarboxylic acid. On the contrary, if so, this is an intramolecular process, and if we are to look at the intermolecular reaction, then we will have COCH3 plus O minus X, and then the hydrolysis of this is going to give me 2 equivalents of acetic acid. So, compared to the intramolecular, the intermolecular gives 2 equivalents, whereas the intramolecular gives 1 equivalent of the dicarboxylic acid. So, if this is K2, then K1 is much greater than K2. That means the intramolecular reaction proceeds at a much faster rate than the intermolecular processes. And in this regard, the first class of compounds which have been actually used as enzyme mimics is cyclodextrins. So, we will first look at cyclodextrins as esterase mimics. And in this regard, the observation is as follows. The observation is that the rate of hydrolysis of paranitrophenyl esters is increased by 7,50,000 by beta cyclodextrin. So now you can appreciate that when we discussed about cyclodextrins, those were an important class of molecules, and now people are trying to use cyclodextrins as biological mimics or enzyme mimics. And the first observation was that the rate of hydrolysis of para-nitrophenyl esters is increased by 750,000 in the presence by complexing it with beta cyclodextrin. So let us look at this process more carefully. So we now have and what has been observed. So we have this O minus here, and we have NO2, COMe, and this is the rate constant in the presence of cyclodextrin, and it gives the corresponding. So I am not going to draw the tub shape every time, I'll just make it simple. So it is going to give me the R, where R is equal to Me plus, so this is what is formed. So we have this reaction here, it goes here, it comes back, and this goes out, plus the phenolate ion, and then hydrolysis of this gives the MeCO2H plus my cyclodextrin back, and I compare this now with the it is the rate constant for this particular hydrolysis to give me the OHNO2 plus MeCO2H. So this is now in the presence of water, so this will give me the corresponding cyclodextrin. And now what is observed is that KCD by K is 100 when the cyclodextrin has been employed. So there is a rate enhancement for this particular ester, which is where it is around the ratio is around 100. So how does this reaction actually take place is what has been shown to you here, that first there is we have the nucleophilic attack of O minus on this ester moiety, and then it forms the OCOR group, and then there is hydrolysis to give you the final product. And during the course of this reaction, what happens is that this particular species is when it reacts, so we need to have this. So you have the O minus here, and now you have got the COOR, and this is so the nitro group is here. You can see here that this is a constrained situation, because the ester carbonyl group is in the plane, and you have the O minus this side, and now there has to be a nucleophilic attack from O minus at the carbonyl center. So it has to approach from the back to the front, hit the carbonyl carbon, and then form a tetrahedral intermediate. This entire thing is taking place within the cyclodextrin. Okay, so it forms this kind of a intermediate. So this tetrahedral intermediate is formed, and now what has to happen is that there has to be the transfer of the acyl group. So there has to be a transfer of the acyl group now. So this is going to form now OCOR and release the O minus NO2. Okay, so this particular process has to take place now. So we now have the acylation which is happening here, and when the acylation happens, then the para-nitro group will be now released actually. So we see these interesting processes taking place. So now what has been observed is that this step is closely related to the action of serine proteases enzymes, in which an acyl group is transferred to serine OH in the first step. Okay, it is transferred to serine OH in the first step. Okay, so we can see this. I think there is a mistake. There is a O here. There is a O here. So now when it comes here, it will go like this. So we'll have to O minus. There is oxygen here. So the step is closely related to the action of serine proteases, in which an acyl group is transferred to serine OH in the first step. So you can see this kind of a rate enhancement when cyclodextrin has been used. The second example I would like to focus on here is example 2 is adenylate cyclase phosphodiesterase activity by cyclodextrin complexes. So let me write down the chemical scheme for this process first. So we have the O minus, P double bond O, O minus, and this is connected to so we have this 5-membered ring here. A is the adenine, this is the adenine part, this is the sugar part. So we have the OH, okay, and this will give first in the presence of beta cyclodextrin and promethium 3 plus, which is a lanthanide. It will first attack here and then form, so it will form this particular product O P OH O minus and double bond O. So it will form this 1, 2, 3, 4, 5, 6, 7 membered ring. 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6 membered ring. 1, 2, 3, 4, 5, 6 membered ring. It will form that, and then here we have the OH. This is the adenine here. plus P2O7 4 minus. So, and this species is referred to as cyclic adenosine monophosphate, CAMP. And so, this is the first stage of the process, and then we are going to have the step 2, that is the second step is the hydrolysis process, which is done in the presence of gamma cyclodextrin and cerium plus 4. So, this is step 1, this is step 2, this gave the product, I am labeling it as A, and this gives the product I am labeling as, after the hydrolysis, O minus O minus. So, this is the product B. So, so what happens is that this AMP molecule, CAMP molecule, is actually the cell to cell, which is called the second messenger in biology. And there are 2 steps, as you can see here. The step 1 catalyzed by adenylate cyclase, and the second step is catalyzed by phosphodiesterase. So, when the cell receives a stimulus, and that is the first messenger, whenever the first stimulus is received, then the compound A is formed, and this compound A is called the which is called CAMP. This compound is formed from ATP, adenosine. This is the ATP, adenosine triphosphate. So, what now CAMP does, it activates other intracellular enzymes to produce a cell response. And in the step 2, this response, which was triggered by CMP, this process is terminated by the hydrolysis of CMP by phosphodiesterase, which is a phosphate ester hydrolyzing enzyme. So, these are the 2 important steps which take place, and both these steps now are mimicked in a synthetic fashion as follows. So, the step 1 is mimicked by a combination of beta CD and PR3 plus, and the second step is catalyzed by gamma CD and C4 plus. And what has been and this is because the cyclodextrin along with the lanthanidine is a hard oxophilic species that catalyzes the overall process. That is what has been proposed, that CD along with this lanthanide have these hard metal oxygen interactions, and overall the species catalyzes the step 1 and step 2, and the results are surprisingly interesting. That is, at physiological pH, and at 30 degrees centigrade the half life for conversion of A to B is only 6 seconds, which is compared to the uncatalyzed reaction, which is 30,000 years. So now the time has dropped to only 6 seconds compared to the uncatalyzed half life of 30 lakh years. So this process of hydrolysis, which is the key step as well, takes place in very short time, and this is what was achieved now at physiological pH and at room temperature. That means the hydrolysis, if you go back to the previous step, that is the termination of this activation process is done by the hydrolysis of CMP by this phosphate ester hydrolyzing enzyme, which now takes place within only 6 seconds, and therefore enhances the efficiency of the process. And we will take another example to understand the relevance of biological mimics, and here we will consider again cyclodextrin as glyoxalase mimics, which involves the isomerization of alpha ketoaldehyde to hydroxy acids, and this is through the involvement thiolate moiety. So let us see how this process, and essentially this process is mimicked by a beta cyclodextrin having an amino ethane thiol group on its primary phase. So let us see this process now. So we have now got a cyclodextrin, okay. We have now got this process here. So here we have got so this is my fuse system, this is my CO. So this is my alpha keto aldehyde, and there is a reaction with here, and this forms the O. Now this comes from here, so this is CO O minus SH. So we have CH2 SCH2CH2N H. Okay, so we have this particular species here. So we can now write it as H, and we have this particular species which is formed. So, this is now my 2-naphthyl glyoxal. Okay, so we now have the cyclodextrin. You can see here it is now positioned here, and this is the primary phase. And now what we can have is we can have the abstraction of the proton here. So it will go like this. This will be taken up here. Oh, sorry. Okay, so it will form now. So we have this reduction now which has happened, and then in the next step we'll do a water wash, and we will take out the corresponding and this is the hydroxy acid. Okay, so now we can see that this is the very important role which is played here by this particular cyclodextrin in catalyzing this particular reaction. So again we can see that cyclodextrin can function as glyoxalase mimics, where it can actually lead to isomerization of this ketoaldehyde to the corresponding alpha hydroxy acid. So overall we see in this particular lecture that we are able to design enzyme mimics which can bring about rates of chemical reactions by templating them at very specific sites on cyclodextrin, which mimic enzyme activity and achieve unprecedented rates, achieving the desired reaction in lesser time, and the product which is formed is also formed in high yields. So with this we come to the end of this lecture, and in the next lecture we will take up more further studies on related systems. Thank you.